All right, guys, welcome back to Underground Science. And in this video, we're going to be talking about toll-like receptors 1 and 2 and examples of microbes that can activate these toll-like receptors. Okay, so toll-like receptors 1 and 2 are actually on the surface of certain like white blood cells, for example, like your dendritic cells or your macrophages, right? And they're on the cell surface. Okay, and what can happen is, so if we have toll-like receptor 1 here, and then we have toll-like receptor 2 here. The reason why I'm drawing it different is because they're heterodimers once they get activated, right? So they're two different proteins, but they dimerize once activated. Okay, so they can dimerize once they are activated. Okay, so we're going to cover certain examples of microbes that can activate these toll-like receptors. So what we should know is that toll-like receptors 1 and 2, right, mainly recognize bacterial lipoproteins. All right, so they recognize bacterial lipoproteins. And the examples of the bacteria we're going to talk about that have these bacterial lipoproteins are Mycobacterium, myco, that's definitely not spelled right, Mycobacterium tuberculosis, and Borrelia burgdorferi. Borrelia burgdorferi. Okay, these two bacteria, right, we're going to talk about as examples, can activate our toll-like receptors 1 and 2, okay, because they have bacterial lipoproteins. Okay, so let's see how this works. So let's talk about Mycobacterium tuberculosis. All right. In Mycobacterium tuberculosis, let's just go step by step. All right, so let's say you would inhale the Mycobacterium tuberculosis. Okay, so that goes to your alveoli. So first we'll represent that in like this purple color. Okay, this will be our Mycobacterium tuberculosis. All right, and you inhale this. To actually do this in a different color, you inhale this, and it goes to your lungs. All right, goes to your lungs, specifically your alveoli. And we know that in our alveoli, we have alveolar macrophages. Okay, so we have alveolar macrophages in our alveoli. Okay, so let's say this is our alveoli right here. And in here, we have alveolar macrophages, which we'll draw in blue. So let's say we have our alveolar macrophage like this, right? And then you have your Mycobacterium tuberculosis that has reached your alveoli. So what's going to happen, obviously, is your macrophage, right? Your alveolar macrophage will try to phagocytose this microbe or this pathogen. Okay, so it's going to try to phagocytose it and engulf it. All right, so let's draw that. So it's going to try to this. I'm just representing this right as engulfing. Okay, it's going to try to engulf this microbe. All right, so that will lead to phagocytosis. So let's draw our macrophage like this, and with our pathogen inside. All right. It's going to try to do that and engulf the Mycobacterium tuberculosis. Additionally, though, since we're not focusing on the um, the pathology or pathophysiology of tuberculosis, we're focusing on toll-like receptors, right? We will talk about tuberculosis and how it works later when we talk about the playlist on our lungs, right? The physiology and pathophysiology of our lungs. But right now we're talking about toll-like receptors. So on the surface of our macrophages, we have toll-like receptors 1 and toll-like receptors 2. Okay, that's what we're talking about in this video. So let's draw them different, right? So in yellow will be 1 and the orange will be 2. So what happens is Remember, toll-like receptors 1 and 2, when activated, will dimerize. Who activates them? Bacterial lipoproteins, which are present on what bacteria? In this case, we're talking about Mycobacterium tuberculosis. So Mycobacterium tuberculosis, right? If we just put that here, so remember, this is representing the Mycobacterium tuberculosis. This has lipoproteins, lipoproteins, right? So the lipoproteins can be released by this Mycobacterium tuberculosis bacteria. So let's say we have that right here, right? This is our bacteria, Mycobacterium tuberculosis, and it's going to release lipoproteins, which will draw like squares, okay? So let's say these are our lipoproteins that the Mycobacterium tuberculosis releases. These can bind onto the toll-like receptors 1 and 2, which will lead to the dimerization of toll-like receptors 1 and 2. So if we draw that, we're going to have the dimerization, right, of toll-like receptors 1 and 2. So I'm just going to draw it simply as like this, right? This is going to be toll-like receptor 1, and this will be toll-like receptor 2. Okay, they're dimerized now, which means that we have activated the macrophage, okay, an intercellular pathway. What is that intercellular pathway? Once toll-like receptor 1 and 2 dimerizes, you activate a signaling pathway inside the macrophage. This signaling pathway is called the MyD88 signaling pathway. Okay, so the dimerization will activate. This is inside the macrophage. Okay, so it's going to be the MyD88 signaling pathway. Okay, let me actually make that better. The MyD88 signaling pathway. What this does is it activates a transcription factor called nuclear factor kappa B. Okay, nuclear factor kappa B. So again, this is going to lead to the activation of a transcription factor called nuclear factor kappa B. Remember, this is all happening inside your macrophages, even though I'm drawing it outside, okay? Because I'm trying to write the steps out. Because we don't have room in this macrophage, but realize this is all happening inside your macrophage. Okay, so then you're going to have the activation of nuclear factor kappa B. Nuclear factor kappa B will then go inside the nucleus of the macrophage. Okay, so then it's going to go inside the nucleus of the macrophage, and it's going to promote gene expression. So transcription and translation. What will that lead to? That's going to lead to certain molecules being produced. So then that'll lead to the macrophage producing, right? That'll lead to the macrophage producing certain cytokines, like tumor necrotic factor alpha, right? So TNF alpha, interleukin 6, interleukin 1, right? A bunch of these cytokines, interleukin 12, right? So what's going to happen is interleukin 12, remember, right, helps the formation of helper Th1 helper T cells, right? So Th1 cells. So basically, remember that once you have a naive CD4 cell, right? A naive CD4 cell, CD4 positive cell, T cell, right? And what if that can differentiate or that can turn into a Th1 subtype or a Th2 subtype or a Th17 subtype. There's many subtypes, but certain cytokines promotes those different subtypes. So for example, if you're exposed to interleukin 4, right, then this naive CD4 cell can turn into a Th2 subtype, Th2 T cell. If this naive CD4 cell is exposed to interleukin 12, then it can turn into a Th1 subtype. if it's exposed to something like interleukin 6 or interleukin 23, it can turn into Th17, the Th17 subtype, right? So what do we say here? So this, if it's exposed to interleukin 4, it can turn into a Th2 subtype. If the naive CD4 cell's exposed to interleukin 12, let's say, it can turn into a Th1 subtype. And if it's exposed to interleukin 6 or interleukin 23, the naive CD4 cell can turn into a Th17 subtype. Now that we know that, right, let's talk about what happens. So again, remember, toll-like receptor 1 and 2, right, responds to bacterial lipoproteins. These bacterial lipoproteins can be seen on Mycobacterium tuberculosis. The Mycobacterium tuberculosis will bind onto the toll-like receptors 1 and 2, right? Specifically, what? What are these representing? The lipoproteins on the Mycobacterium tuberculosis. Those lipoproteins are released by this bacteria that binds to toll-like receptors 1 and 2. Toll-like receptors 1 and 2 then dimerize, activating the MyD88 intracellular pathway. MyD88 activation leads to the activation of nuclear factor kappa beta, or kappa B. I apologize, kappa B. Then nuclear factor kappa B goes into your nucleus and induces gene expression, leading to the production of certain cytokines. What are those cytokines? Tumor necrotic factor alpha, TNF alpha, IL-6, IL-1, IL-12. And IL-12, as we said, can turn a naive CD4 T cell into a Th1 subtype. Remember, the Th1 subtype releases things like interferon gamma and interleukin 2, right? So interferon gamma will do what? It's going to activate macrophages, right? So this will release interferon gamma. This will activate macrophages, and remember, it's going to help also start the formation, or it's going to aid in the formation of a granuloma. Okay, help with formation of granuloma, because remember, once interferon gamma is released, it activates the macrophages. Interferon gamma then also helps epithelioid histiocyte formation, right? Giant cell formation, which is basically the nuclei of many macrophages coming together. And then, of course, all these cytokines will attract more T cells, which will be part of the granuloma formation, right? And actually, since we're already here, tumor TNF alpha, right? What it does is it actually promotes granuloma formation. Okay, so this also, let's draw this also. This also promotes granuloma formation. Remember, Mycobacterium tuberculosis causes caseous necrosis. Okay, so what that means is inside the granuloma, it's going to be necrotic tissue. Okay, and then of course, interleukin 6 can go to your liver and help increase acute phase reactant proteins. So IL-6 can go to your liver, and your liver can then produce acute phase reactant proteins like CRP, for example. So you'll see that elevated if you do a blood test for that. Okay. So those are the basic things about how toll-like receptors 1 and 2 get activated based on bacterial lipoproteins that are secreted by Mycobacterium tuberculosis. Okay, so we can see that toll-like receptors 1 and 2 are important in fighting off bacteria that release lipoproteins. Because if you had a mutation, let's say, right, in toll-like receptors 1 and 2, then of course you'd be more susceptible to tuberculosis, right? So that's how that works with tuberculosis. But there's another bacteria we should talk about, as we mentioned in the beginning of the video, is Borrelia burgdorferi. Okay, so let's see how Borrelia burgdorferi works in terms of activating toll-like receptors 1 and 2. So let's write that here. Borrelia burgdorferi. As we know, um, this bacteria can cause Lyme's disease, okay? And it's mostly spread by by ticks, of course, right? If the tick bites a patient, it can, if that tick is carrying the Borrelia burgdorferi bacteria, that can lead to Lyme's disease. Okay, so what's going to happen is, let's say a tick injects this bacteria, Borrelia burgdorferi, right, into the skin. Again, remember, pathogens like Mycobacterium tuberculosis and Borrelia burgdorferi have bacterial lipoproteins on it, and they can release those lipoproteins. All right. And if they release those lipoproteins, what's going to happen is we're going to have macrophages or dendritic cells, right? Remember these cells have toll-like receptors on their surface. Again, let's take the example of a macrophage since we've already been talking about that, right? So if we have a macrophage here and we have Borrelia burgdorferi, let's draw the bacteria like this, right, in green, and it releases, again in squares, we're going to say lipoproteins. Now, these lipoproteins, again, right, will bind onto toll-like receptors. So let's say we're talking about toll-like receptors 1 and 2. So we're going to have toll-like receptor 1 here, and let's draw 2 in a different color. These will bind onto the toll-like receptors 1 and 2, causing dimerization. Okay, so you're going to form heterodimers. So this will cause dimerization. So we have our macrophage here, our nucleus. Then we're going to have dimerization. So we're just going to draw it like this. Remember, this is toll-like receptor 1, and the purple is toll-like receptor 2. Okay, so now it's dimerized. Remember what happens after that? We activate the MyD88 intracellular pathway. So first, we get dimerization of the toll-like receptors 1 and 2. Then we get activation of the MyD88 intracellular pathway. This then leads to the activation, remember, of nuclear factor kappa B. Nuclear factor kappa B goes into your nucleus, in the macrophage's nucleus, and does what? Remember, it leads to the production of cytokines. Okay, so what cytokines are we talking about? Remember, it's just generally it's going to produce, and like we said in the previous slide, when we're talking about Mycobacterium tuberculosis, we're going to have TNF alpha production. That's not alpha, right? We're going to have interleukin 1 production. We're going to have interleukin 6 production. We're going to have interleukin 12 production, and so on, right? So, like, what will these cytokines do? So, again, all these cytokines, as we know, can go to the hypothalamus and trigger fever, right? And of course, cytokines induce inflammation. So we're going to have inflammation. And we already talked about interleukin 12, right? How it can help the naive CD4 cells turn into the Th1 subtype, right? Another interleukin that actually I'm thinking of right now that it may be important to keep in mind is interleukin 8, right? Interleukin 8 can also be released due to this nuclear factor kappa B stimulation, right? And as we know, interleukin 8 attracts what? Neutrophils, right? Attracts and activates neutrophils. So neutrophils that will then come to the site, right, where the Borrelia burgdorferi got um, transmitted from the tick in our skin, right? And then that's going to help fight off, obviously, the bacteria. And then, of course, your normal immune system, as we know, the macrophages and dendritic cells will try to engulf it, present it to CD4 T cells. Those is going to lead to cytokine release and hopefully help activate our adaptive immune system, right? The adaptive immunity part, hopefully lead to release of antibodies and all that. But that's going to be in a separate video talking about the immune system, of course. This is just focusing on the role of toll-like receptors, especially 1 and 2. Okay, so that's actually it for this video. In this video, the main goal was to understand that what activates toll-like receptors 1 and 2, and go over certain examples, which we did. Okay, so I hope this video wasn't too confusing. I hope this made a little bit of sense and you guys learned a little bit. And thank you for watching and see you guys in my next video.